Hi, my name is Scott and welcome. This presentation is on proteins, and this picture represents the many different types of foods that contain proteins. Some groups may have more proteins than others, and one may have more complete proteins than the others, as we will soon see. So proteins have very important functions in our body and play diverse roles. So I have a wide range of topics, starting with a general overview of the proteins, amino acids, protein synthesis, the structure of proteins, protein metabolism, the functions of proteins, digestion and absorption of proteins, proteins in foods, health effects of proteins, protein recommendations, and protein energy malnutrition. So the general overview of proteins is our body is made up of thousands of proteins. Proteins contain carbon, hydrogen, oxygen, nitrogen, and some contain sulfur. The functions are they regulate and maintain body functions. They are structural materials, and they provide an essential form of nitrogen, and that's in the form of amino acids. Amino acids are the building blocks of proteins. So amino acids. Basically, all amino acid structure is the same. It starts with a central carbon, and they have an amino group. Now, you may hear this referred to as an amine or an amino. It just depends if it's whether it's a prefix or a suffix. An amine would be a suffix, such as if you have CH3 and then the NH2, that would be methylamine. But for the purposes of this presentation, I'm going to refer to it as an amino group. So again, it will have a central carbon with an amino group. It will have an acid group, which is carboxylic acid, and it will have a hydrogen off its central carbon. Now, what makes each one of the amino acids different from each other is this side group or side chain. And you'll see this in books as an R represents a side group or side chain of carbons. So that separates all the amino acids from each other. So there are 20 different amino acids that make up all the proteins. In our English language, there are 24 letters in our alphabet, and they comprise the whole language. So the 20 different amino acids, they will comprise all the proteins in our body. There are 9 essential amino acids and 11 non-essential amino acids. And then I'll go a little bit further in this presentation and show you a little difference of them being conditionally essential. There are 7 of the 11 that are considered conditionally essential. So here again are the amino acids. The way I learned this is leucine, isoleucine, and lysine. Then histidine, methionine, phenylalanine, threonine, tryptophan, and valine. Now, if you learn these essential amino acids, you got half of it licked if you have a test question, and you'll know the difference between an essential and a non-essential amino acid. So the non-essential amino acids are arginine, asparagine, glutamine, glycine, proline, serine, tyrosine, alanine, cysteine, aspartate, and glutamate. Now, out of this, these are the essential amino acids like I just spoke about. And together, these two columns represent the non-essential amino acids together. However, they will be broken down a little bit in a couple slides where I'll show you the representation of what it means by a conditionally essential amino acid. Okay, essential amino acids, again, they are considered indispensable amino acids. They are amino acids that the human body cannot make in sufficient amounts to meet the physiological needs of the body, and therefore they must be included in our diet. Now let's start with leucine and isoleucine. These are 2 branch chain amino acids because this carbon here has more than 3 carbons attached to it. The same with isoleucine. Leucine and isoleucine are similar in structure, they just have a different conformation. This is an isomer of leucine, so it is the same, it has the same molecular formula, same number of carbons and hydrogens, oxygens and nitrogens. But it simply has this methyl group brought up to here. So it is pretty much the same. These are 2 branch chain amino acids, and valine is also a branch chain amino acid. It has more than 3 carbons attached to a carbon. And lysine has a carbon chain that is called aliphatic. If it has a carbon chain or a lone carbon chain with the branched carbons off of it, it's considered aliphatic. So lysine has another amino group. Histidine, it is aromatic. It's called aromatic because in the past these rings would give a sweet smell. So it has a ring structure with a double bond every other carbon, and that's considered aromatic. Methionine, it is one of two amino acids that has a sulfur group in it, and it is also aliphatic. It has a long carbon chain. And phenylalanine, it is aromatic. It has a ring structure with a double bond every other carbon. And this is a hexene, hex meaning 6, and ene meaning double bonds. And they also call this a benzene ring. So phenylalanine, as you'll see in a couple slides, that this would be converted to tyrosine. Right now it doesn't have a hydroxyl here, but there will be a hydroxyl added to this to turn it into tyrosine. Here we have threonine, and it has a carbon chain and a hydroxyl coming off of that, so it is aliphatic. Tryptophan is aromatic. It has two ring structures that has double bonds every other carbon. And valine, like I said, that is the third branch chain amino acid in our body. Now, non-essential amino acids. These are amino acids the body can make in sufficient amounts to meet our physiological needs. And starting with alanine, this is the second simplest form of an amino acid. It simply has a methyl group attached to its central carbon. And cysteine is the second amino acid that has the sulfur group. Aspartate and glutamate, they are very similar. They both have two acid groups, but glutamate has an additional carbon with its respective hydrogens to meet the carbon bonds. Now these are the non-essential amino acids. Again, these are 7 more, but I've grouped these together to be looked at as conditionally essential. But first, let me go over them as being non-essential. Excuse me. So tyrosine, again, this is phenylalanine, but it has a hydroxyl added to it. And the enzyme that will do this is hydroxylase, but we're not going to go over that right at this point. Asparagine and glutamine are very similar. They both have an acid group, and the glutamine just has an extra carbon added to it with its respective hydrogens. Glycine is the simplest amino acid we have. That has 2 hydrogens bonded to its central carbon. Proline, it is aliphatic, and serine, it is pretty simple looking amino acid. It has a carbon with a hydroxyl hanging off of it. And also arginine, it is a aliphatic amino acid, meaning it has a carbon chain, and it has 4 nitrogen groups, 1, 2, 3, 4 nitrogen groups. Conditionally essential are amino acids that are normally non-essential, but must be supplied by the diet under special circumstances when the body's need for it exceeds its ability to produce it. Now, we discussed the phenylalanine a second ago, having the benzene ring, the aromatic ring, and here we add the hydroxyl to it, and it converts it to tyrosine. Now, what makes this non-essential right here is we had the enzyme to convert this to tyrosine. But now, phenylalanine is essential, and tyrosine is non-essential because we can convert phenylalanine to tyrosine. But if we are diagnosed with phenylketonuria, we don't have that enzyme hydroxylase, so we can't convert phenylalanine to tyrosine. And the essential amino acid phenylalanine, like I said, we can't convert it, so tyrosine now becomes an essential amino acid because we can't produce it from an essential amino acid. Okay, again, I was telling you about the branch chain amino acids. Each one of these, leucine, isoleucine, and valine, are branch chain amino acids because they have more than 2 carbons attached to a carbon. So each one of these have 3 carbons attached to it. And this has 3 carbons. Valine has 3 carbons attached to it. Now, if you look at the difference between leucine and isoleucine, basically you just have a rearrangement. Iso means kind of the same. It's just a different conformation. So it has this methyl group put up here. And basically, this will come down. So you have pretty much the same, you do have the same molecular formula, the same number of carbons, and the same number of hydrogens. But it is just, its arrangement is different, so that's why it's an isomer, isoleucine. And this is valine, like I was saying. So these are the 3 branch chain amino acids, and they are the primary amino acids used by muscles for energy, and they comprise approximately about 35% of the amino acids in our muscles. Okay, protein synthesis, excuse me. Okay, I went over transcription and translation in the digestion and part of my lecture. So here again, I'll do it again. Transcription is a process of copying DNA in the nucleus, and that DNA is deoxyribonucleic acid in the nucleus to make RNA, ribonucleic acid. An RNA polymerase binds to the promoter site. Now this is this light blue thing back here is an RNA polymerase, and that will bind to this promoter site. And what that does, once it binds there, it will work its way down this double-stranded DNA helix and separate it. So as it separates it, each unpaired base will bind to a complementary nucleotide binding to a messenger RNA strand. So this nucleotide bases will have a complementary nucleotide bind to it and start building this messenger RNA strand. The process stops when the RNA polymerase reaches the gene's termination point. So it will go down here. This RNA polymerase will work down this DNA double helix, separating it, and it will stop when it comes to a specific termination point. In that gene, genes are specific segments of DNA, and they have a promoter site and a termination point, and to where it will build a messenger RNA, and then it will be transcribed into a protein. Translated into a protein, sorry. Translation. It is the process of translating the messenger RNA into a protein. In the ER, the endoplasmic reticulum, translation begins with the binding of a messenger RNA to a small subunit of ribosome. Now, this is a small subunit of ribosome, and there's a larger one that comes along. But this small subunit of ribosome, a messenger RNA, will exit the pores of the nucleus, go into the ER, the endoplasmic reticulum, and bind to the ribosomes. At the endoplasmic reticulum, there is the rough ER and the smooth ER. If you see the grainy surface on the rough ER, those are ribosomes. And those ribosomes are protein assembly plants, if you will, kind of like the Ford Motor Factory. They build cars. Well, the ribosomes build proteins. And so this messenger RNA will bind to this ribosome, and that will initiate a signal for a transfer RNA to come down here and bind to this messenger RNA. Now, this transfer RNA, it has 3 nucleotide bases, and that is called the codon. 3 nucleotide bases is called the codon. And that transfer RNA will have its specific protein with respect to its codon. Now, that tRNA will come down and bind to this messenger RNA. And the binding of that messenger RNA, like I said, in translation, there are three parts. It's initiation, elongation, and termination. And like I said, the binding of this messenger RNA starts the initiation, the transfer RNA complementary codon binding to messenger RNA with an attached amino acid. Then a larger ribosome attaches to the group that has 2 binding sites, two binding sites of ribosomal proteins and ribosomal RNA. Then what happens is this larger ribosomal subunit will come in and bind to the smaller subunit, sandwiching the messenger RNA in between there. Then another transfer RNA will come down and bind to this messenger RNA. And then the proteins will start to bind together, and that is called a peptide bond. The binding of the amino acids together leads to elongation of amino acids forming proteins. Once the ribosomal subunit reaches the termination point, it separates, releasing the newly formed proteins. And if you remember in the human anatomy and physiology section of one of my presentations, the proteins will be excreted into the Golgi apparatus, where they are sorted, packaged, and sent out of the cell, used in the cell or sent out of the cell to be used. Okay, polypeptides. A peptide bond connects the acid end of one amino acid to the amino end of another amino acid, forming a peptide bond in a protein chain. Let me show you what I'm talking about. This is an amino acid, excuse me. This is an amino acid. It has the amino group, it has the hydrogen off the central carbon, and it has the acid group. This is a carboxylic acid. Now, what I was saying is the acid group of one amino acid will bond to the amino end of another amino acid. So the carboxyl group will lose an oxygen and the amino group of the other amino acid will lose 2 hydrogens. Together, that is H2O, the leaving group will be water. And then that will form the peptide bond. Okay, a dipeptide is 2 amino acids together. Tripeptide is 3 amino acids bonded together. And a polypeptide is 10 or more amino acids, that are bonded together. Okay, sequencing of amino acids. Polypeptide chains can twist into a variety of shapes depending on the amino acid sequences. Like in our alphabet, specific letters will form words, and those words have a function in our language. The same is with proteins. Each amino acid in sequence with each other will lead to a specific function. The unique side groups of each amino acid can give characteristics that attract it to or repel it from the surrounding fluids and other amino acids. Side groups may have electrical charges that make the amino acid hydrophilic, water-loving, or hydrophobic, water-hating, leading to a conformational shape. The ends of polypeptides, chains, or other parts of amino acids may be attracted and cause the chain to fold, taking stable shapes. The order of the amino acid sequences in a protein determines its ultimate shape, and the protein's final shape will determine its function. So the structure of proteins. Basically, there are 4 structures in proteins. There is the primary structure, and that is the sequence of the amino acids. Say it's cysteine, phenylalanine, tryptophan, and valine. Those are a specific sequence, and they will determine an amino acid shape dependent upon the bonds and the interactions within each other. The secondary structure, that is where it forms as the electrical interactions within the polypeptides. So the negatively charged hydrogens and the positively charged oxygens, say for example, if you see this here, the amino acid, it has the acid group and the amino group, and then the hydrogen and the oxygen will start to try to bond together, making this into like a pleated sheet, a beta pleated sheet, or an alpha helix. That's the secondary structure. The tertiary structure, it forms as the side groups interact with their environment and to each other, forming convoluted twists and folds. So it starts to be twisted, convoluted in many different folds, and that is the tertiary structure. The quaternary structure is where the interactions of 2 or more polypeptides form globular proteins. And this is 2 or more of these tertiary structures. So it becomes a globular protein, as we will see in hemoglobin. So human insulin. Insulin is composed of 2 polypeptide chains. One chain has 21 amino acids. This is the small chain, and this is the larger chain. The small chain has 21 amino acids, and the larger chain has 30 amino acids. And when I was talking about specific amino acid sequences to form a shape and a specific function in a protein, for example, here, the small chain, and this insulin will always have this specific sequence of amino acids. So this is glycine, isoleucine, valine, glutamate, glutamine, cysteine, threonine, serine. So we'll always have that specific amino acid sequence. Now the short chain has a disulfide bridge, has a sulfur connecting the cysteine. So that will draw that polypeptide in. And also the ends will also interact, and the hydrogens and oxygens between the amino acids, they will interact, all forming a specific shape. So also there are, in the long polypeptide, the long chain, 30 amino acids, it has 2 disulfide bridges connecting it with the small chain. So its conformational shape is determined by its amino acid sequences, the electrical interactions between them, and the 3 disulfide bridges. Same with hemoglobin. Excuse me. Okay, hemoglobin, it is a quaternary structure. As you see, there are 4 convoluted twists and folds of the tertiary structure becomes a quaternary structure. It contains 4 polypeptide chains that make up the red blood cells. Hemoglobin carries oxygen from the lungs to the cells throughout our body. Now, each one of these polypeptide convoluted chains, they have a heme group, an iron group, which is a mineral, and that allows hemoglobin to function, and that will allow the oxygen to bind to the iron. So again, a little more with amino acid sequencing. The sequences of amino acids determines the function of a protein. Genetic errors or mutations in sequencing can lead to significant consequences. In sickle cell anemia, 2 of hemoglobin's polypeptide chains have an altered sequence of amino acids, leading to an elongated crescent or a sickle-shaped cell, blood cell. The change in hemoglobin's shape decreases its ability to transport oxygen. Now, the normal sequence of amino acids in hemoglobin is briefly here. We have valine, histidine, leucine, threonine, proline, and glutamate. But the problem here is that the glutamate, there is a genetic error or mutation where valine is substituted. Now, with valine, this sequence of amino acid, it will change the shape of the protein because of the electrical interactions amongst the amino acids and the side groups with its environment. So it will change the shape of the amino acid to a sickle shape. It changes the shape of the polypeptide. And that's how we get sickle cell anemia. And that will decrease its ability to transport oxygen because its shape determines its function. So protein metabolism. Protein turnover. That is, proteins are continually synthesized and catabolized in our body. They're always built and broken down. We don't store proteins in our body. We use proteins, amino acids, to build amino acids. And no proteins are synthesized for life. They are continually degraded. And adults degrade 1 to 2% of their body protein daily. Around 75 to 80% of the freed amino acids are reutilized for new protein synthesis. And you don't have to memorize these two numbers, the 1 to 2% and the 75 to 80 at this level. I'm just trying to give you a general idea of the protein turnover in our body. Now, there's what we call an amino acid pool. These are all the amino acids in your body and the ones that you consume in dietary intake. Let me give you the kind of definition of the amino acid pool. Is the free amino acids in the body and the ingested dietary protein that are catabolized to amino acids that can be used to make new proteins, other compounds, or be used as energy constitutes the amino acid pool in the body's cells and throughout our blood. So a little bit more about protein turnover. And here I want to start relating some of this protein turnover to nitrogen balance. So for example, if we ingest approximately 90 grams of protein is ingested daily, and approximately 70 grams of protein per day enters our digestive tract through gastric and intestinal juices. Digestive enzymes and cells sloughed off from the gastrointestinal lining. So all of those proteins and amino acids that are broke down, that comprises approximately 190 and 70 is about approximately 160 grams of protein enters our digestive tract daily. And there's no need to memorize these numbers. This is good to know, though. This is in the bachelor level. But out of this 160 grams, you'll lose 10 grams worth of nitrogen. Now, I want to give a relationship. Protein is 16% nitrogen. So 10 grams of protein, if nitrogen is 16% of protein, then you'll simply go 0.16 times 10 grams, and that will give you 1.6 total nitrogen that is excreted in the feces. So we lose approximately 10 grams worth of nitrogen in our feces, and that is why we will catabolize to amino acids and small peptides approximately 150 grams of protein. Now let me show you what I was talking about. Say, for example, we take in, again, about 90 grams worth of protein. Now this, I am doing protein turnover and nitrogen balance here at the same time. Protein intake is about 90 grams, and we bring into the gastrointestinal tract through gastric and intestinal juices and gastric enzymes, and the cells sloughed off from the gastrointestinal lining. All those proteins will be broken down to amino acids, and that will leave about 150 grams of protein are absorbed into our body. Now, out of that, I said about 10 grams worth of nitrogen will be excreted in the feces. Also, we will excrete about 75 grams of protein worth of nitrogen. So if you did the 16% of 75, you'd come up with 12 grams of nitrogen. And again, other losses such as sweat, hair, nails, will also leave about 5 grams worth of nitrogen, and 16% of 5 grams is 0.8 grams of nitrogen. So the total, again, what I'm showing you here is the nitrogen balance. The protein that we take in has 16% of nitrogen, and that is equal to 14.4 grams worth of nitrogen. So nitrogen balance is the protein taken in and the nitrogen losses, and it is a balance. Nitrogen balance. Nitrogen balance is the difference between total nitrogen intake and total nitrogen loss in the feces, urine, sweat, hair, nails, etc. Normally healthy adults are in nitrogen equilibrium. Nitrogen intake equals nitrogen output. Positive nitrogen balance, that means you're taking in more protein than you're excreting in the amount of nitrogen. Nitrogen intake is greater than nitrogen excreted. It is characterized in growing infants and pregnant women, bodybuilding adults, endurance athletes, and when there is tissue growth or replenishment, such as recovering from metabolic stress or a nutritional deficiency. So you're increasing your nitrogen intake. Negative nitrogen balance, that's when you're not taking in enough proteins and you're excreting more nitrogen. So nitrogen intake is less than nitrogen excreted. And that's seen in fasting or starvation when there is no or inadequate protein intake. After surgery, in advanced cancer, in pathological conditions such as burns, traumatic injury, fever, and in severe psychological stress. So transamination. This is transferring an amino group from an amino acid to a keto acid, forming a new non-essential amino acid and a new keto acid. Now, what do I mean by this? Basically, what I was saying earlier is protein turnover. We continually break down and build amino acids and proteins in our body, and we don't store proteins, so we'll convert one amino acid to another. Say, for example, if we have plenty of cysteine, our body has plenty of cysteine, and our dietary intake didn't allow us to get enough alanine, for example. This is just an example, alanine. So let's say, for example, this amino acid has a side group of cysteine, and this amino acid, actually this is a keto acid, it has a ketone here, and it doesn't have the nitrogen group. So this is an amino acid. It has the acid group, the amino group, the hydrogen, and the side group, and here I'm just saying, for example, it has the cysteine side group. And this side group here would be an alanine side group, but it's not an amino acid, it's a keto acid. So if our body has plenty of the cysteine, but we need alanine, it will simply take this nitrogen group and put it onto this keto acid, turning it into a new amino acid. And now we have alanine in our body because we're not taking in enough alanine, or basically alanine is used a lot in our muscles, so we're not producing that. So now we have a keto acid and an amino acid. Deamination. This is where an amino acid loses its amino group and hydrogen atom, forming a keto acid and ammonia. So here we go. We have an amino group. This is an amino acid altogether. We have the amino group, the acid group, the hydrogen, and the side group on the amino acid. Now, to break this down, what we're going to do is basically this is how we get rid of ammonia in our body, through urea. So this is deamination. We're going to take the amino group off. We're going to deaminate it. And we do that by a couple processes. We either do it through hydrolysis or a reduction. So we'll don't need to know that. But basically, we'll take this nitrogen group off and the hydrogen, and we'll have ammonia. The carbon skeletons from the amino acids can then be used for energy or for the production of glucose, ketones, cholesterol, or fat. And I have a slide coming up to show you, to depict what I just said. to be able to use this carbon skeleton. Now, once we take this amino group off of it to get rid of the ammonia, then we can use this carbon skeleton. You know, these are like Legos, these carbons. You know, you can kind of refer to it as Legos. You just keep building carbons. You can use them for energy and stuff, and I'll show you how that works out. So, like I was saying, we get rid of ammonia through the urea cycle. The urea cycle is primarily in the liver and to a lesser extent in the kidney. The urea cycle eliminates two metabolic waste products, ammonia and carbon dioxide, in the form of urea. Now, we'll take two ammonias and add that with a carbon dioxide. And we'll take the hydrogen from one ammonia and another ammonia and an oxygen off of there. And again, the leaving group is water. So two ammonias and one carbon dioxide, two oxygens, will leave us with urea. And then we'll excrete the urea as urine. So basically what happens is the amino acids come in, we deaminate them, and the nitrogen will bond with the carbon dioxides, and then that will form urea. The blood will go through the kidney, and it will filter the urea out and be excreted as urine. So as dietary protein increases, urea production increases. The liver releases urea into the blood and the kidneys and excrete urea as urine. Okay, a little more about protein metabolism. Basically here, I'm going to go through this pathway. It's not very complicated. It's kind of fun. I like this kind of stuff. But basically here, I just want to show you that amino acids can be converted, and in this case, tyrosine can be converted to neurotransmitters. The neurotransmitters being norepinephrine and epinephrine. So these neurotransmitters, they're hormones that are released from the adrenal glands, leading to the fight or flight response that builds up for excitement, and of the sympathetic nervous system. And what that does is it increases blood glucose. It increases our energy in the bloods that we may need it to fight or flight. So let me briefly go over this pathway. Again, tyrosine will be converted to neurotransmitters, norepinephrine and epinephrine. This is norepinephrine, epinephrine, and tyrosine. And we get through this through not too many enzymatic processes. First, the tyrosine will get a hydroxylase. Remember I was telling you about phenylalanine. We'll get a hydroxylase. We'll put a hydroxyl on phenylalanine and turn it into tyrosine. But we're starting with tyrosine, and we get a hydroxylase, and we simply add another hydroxyl to it. Pretty simple. Then we get a decarboxylase. We carboxylase. We'll take off this acid group right here. We take off the carbon. Decarboxylase will take off the carbon, and we'll be left with dopamine. 2 steps of dopamine, and then we get another hydroxylase, and now we'll put a hydroxyl right here, leaving us with norepinephrine. And simply we do a methyltransferase, and you don't need to know all these enzymes. Basically, again, tyrosine will be converted to neurotransmitters. And then you put the methyl on down there, and you have epinephrine. Also, tyrosine can be converted to melanin. Melanin is a pigment in the skin, hair, and eyes that results in a red to brownish coloring. And this is a multi-enzymatic process. I'm not going to go over this because it does become very involved, and we're not at that step. But I just want you to recognize, again, that tyrosine can be converted to melanin also. And this is tyrosine. It will go through several steps, dopaquinine, several steps, and then finally to a multi-ring structured melanin. Okay, another one, tryptophan. Tryptophan can be converted to serotonin and niacin. Serotonin is a neurotransmitter involved in sleep regulation, appetite control, and sensory perception. And tryptophan to serotonin, it is basically 2 quick steps. Let me just go over that briefly. You have tryptophan, and again, you have a hydroxylase. You add a hydroxyl onto this benzene ring, and there it is, and you have serotonin. Now, going to tryptophan to niacin, it is more involved, and this is a multi-enzymatic process that will lead tryptophan to niacin. And niacin deficiency, that leads to pellagra, and it's characterized by the 4 Ds: diarrhea, dermatitis, dementia, and death. And also, niacin is it has been shown to increase high-density lipoproteins, which in turn will decrease cardiovascular disease risk. Okay, tyrosine can also be converted to thyroxine. Thyroxine, that is a hormone produced by the thyroid gland that regulates metabolism. Iodine is necessary to convert the T3, this is T3 to T4. I'll go over this briefly in a second. T3 to T4 in iodine, a deficiency in the iodine will lead to goiter, and that will lead to an enlarged thyroid gland that you can visually see. So tyrosine, basically this is kind of simple, but again, there's no reason to memorize these pathways. Just recognize that tyrosine can be converted to thyroxine, which regulates our metabolism. Now, here simply we have tyrosine. This is the benzene ring with a hydroxyl. We went over that several times. And here we just add another benzene ring to this and 3 iodines. And that will make, let me see if I can get this right, triiodothyronine, and that's T3. So in order to convert T3 to T4, we need another iodine. And that's where it was saying iodine is necessary to prevent goiter. So again, we have the amino acid metabolism. Tyrosine can be converted to thyroxine. Okay, the functions of proteins. Excuse me. Okay, proteins are involved in growth, repair, maintenance, and replacement of injured and worn out tissues and cells. Proteins facilitate and regulate processes in the body and form parts of many body structures, including bone, muscle, blood, and skin. Keratin and collagen are the most abundant proteins in the body. Keratin is the main protein in the skin, hair, and fingernails, and collagen is used as building material for ligaments, tendons, blood vessels, and to form scar tissue. Protein provides the most satiety of the 3 energy-yielding nutrients. Now, this could go into a long, drawn-out explanation, but for this point right here, just recognize that protein gives you the best feeling of fullness out of carbohydrates and fats. Proteins also function in fluid balance. Under normal conditions, blood pressure forces water into capillary beds and into extracellular spaces. This is the arteries bringing in the blood supply to the capillary beds, and the capillaries are just vessels with a single cell wall thickness. And the nutrients will drain into these capillary beds and then drain into the tissues and the cells to be nourished, and the fluids will drain in between the interstitial spaces, in between the cells. So since proteins are too large to diffuse across capillary walls, they remain in the blood vessels and attract the water back into the blood vessels, creating a balance between blood pressure. So we have the blood pressure pushing the fluid into the capillary beds and into the tissues and the interstitial spaces. And as the proteins start coming back through the venous capillaries, the veins, they will draw the water back into the veins. But with inadequate dietary protein intake, excessive protein loss, and decreased protein synthesis, the water is not attracted back into the blood vessels, causing a buildup of fluid in the extracellular spaces, leading to edema. And this is a picture of edema. What happens is the water is not attracted back into the venous capillaries, and it starts building up in the subcutaneous tissue, the fat tissue right below the skin, and it starts to leave a swelling, and that's called edema. And if you put your thumb in it, it will leave an indentation, and that's also referred to as pitting edema sometimes. So proteins also function in acid-base balance. Blood pH has a very narrow range excuse me, that falls between 7.35 and 7.45. A blood pH that is below 7.35 leads to acidosis, and a pH above 7.45 leads to alkalosis. Excesses in either direction, less than 7 and greater than 8, can result in coma or death. The body undergoes many metabolic processes that exchange ions. Hydrogen ions have a positive charge, and increased hydrogen ion concentrations create an acidic environment. Proteins have a negative charge that accepts the hydrogen ions, thereby acting as buffers to maintain blood and cellular pH. So the proteins will attract these hydrogens and thereby decreasing the acidic environment. Proteins also form structural channels in cellular membranes that act as pumps. They pump hydrogen ions across the membranes to maintain a cellular ion concentration and maintain the blood to a slightly alkaline pH. And a balanced pH is 7, but since our blood is 7.35 to 4.45, it's slightly alkaline. Okay, proteins also function as enzymes. This is a picture representation of an enzyme here. Almost all enzymes are composed of proteins. Generally, they are three-dimensional globular proteins. Now, this is a three-dimensional globular protein. And say, for example, if you have binding sites on these enzymes for substrates, and let's say, for example, let me just pick one at a random. This green part right here could be a binding site on this protein. And the shapes determine its function. And maybe these other amino acids around here, all these proteins, will prevent any binding anywhere except for this green spot. So with that being said, enzymes are catalysts that promote one form of the molecule, a substrate, to a different molecular form, a product. So enzymes like this, it will take a substrate, say for example a disaccharide, sucrose. Say for example this is sucrose, and this is a glucose and a fructose, two monosaccharides. Now they will bond to this enzyme, and they have a specific conformational shape that will allow this to bind right to it. And what that will do is it will, in this case, that enzyme will turn this, and that enzyme is sucrase, will turn that into two monosaccharides, a glucose and a fructose. Proteins also function as hormones. Hormones are chemical messengers released from cells or glands, endocrine or exocrine glands, that send signals to other cells. Many hormones are composed of proteins. For example, we spoke about thyroxine, derived from tyrosine, which regulates our metabolism. Insulin is composed of a total of 51 amino acids, and glucagon is composed of 29 amino acids, and they control blood glucose levels. Norepinephrine and epinephrine, derived from tyrosine, increase blood glucose levels. And antidiuretic hormone, ADH, is a peptide hormone that regulates fluid and electrolyte balance. Okay, proteins also function as transporters. Excuse me. There are many different types of protein transporters that have a specific function. For example, hemoglobin, that transports oxygen. We went over that. Lipoproteins, we went over that in lipids. They transport lipids. And there are channel proteins that act with facilitated diffusion. The protein facilitates the diffusion across the membrane, as, for example, in fructose, if we remember that. Also, there are carrier proteins, which needs energy. So that is like glucose and amino acids. So that will bring in the glucose or amino acids, and with energy, it'll transport it across a concentration gradient. Also, there are voltage-gated ion pumps. This is what I was telling you about there are hydrogen pumps that maintain cellular concentrations of hydrogen. And this is a sodium-potassium pump. And it is also done with energy through active transport. Proteins also function in the immune system. Now this is, we're not at this level, we don't have to recognize anything about the structure of an antibody. Basically, I'm letting you know that proteins function in the immune system as antibodies. So let me take it just a little further. Antibodies, they're composed of proteins. The structure of all antibodies are generally the same. They contain 4 polypeptide chains, 2 light chains that contain about 220 amino acids. This is a light chain, and this is a light chain, and this is a heavy chain, and this is a heavy chain, which is twice as long as the light chain. So the light chain has about 220 amino acids, and the heavy chains have about 440 amino acids. Now, this is the binding sites that the pathogens will, the antibodies will bind with the pathogens here, and those are the amino groups. So anyway, like I was saying, proteins function in the immune system as antibodies. Now, if you remember, I was saying that the carbon skeletons from the deaminated amino acids can be used as energy, glucose, or lipids. Well, here we can use these amino acids. These amino acids, this is the citric acid cycle. Earlier I spoke about glycolysis, is turning glucose into pyruvate and then getting it into the citric acid cycle, where it will go around through a number of intermediates generating hydrogens and electrons that will send down to the electron transport system that will generate energy. So we get the pyruvate into the citric acid cycle, and I don't want you to memorize any of this as yet. I'll go over that in the metabolism, our next lecture. But here, basically, we have a couple intermediates. Here I've chosen 5, 6, 7 intermediates in the citric acid cycle. And here are several proteins that come in at alpha-ketoglutarate. You don't need to memorize this, but they're coming in at different points. These amino acids can come in to each one of these intermediates in the citric acid cycle, and this will generate electrons to go for energy. So these amino acids can be used to generate energy. Also, these amino acids can come up to oxaloacetate, which will go to phosphenylpyruvate, PEP, and that will be converted to glucose. So we can generate energy, glucose, and also some of these ketogenic, these are glucogenic, meaning glucose, they will go in to convert to glucose. But these will convert to ketones or fatty acids. So these amino acids, since we don't store proteins in our body, we either use them as structures or maintaining, building, repairing, or we can use them if we have the required amount of proteins and we have excess and we have a sufficient amount of fuel and fats, we will use these as energy or we will store them as fats. Now these amino acids will go to acetyl-CoA or acetoacetyl-CoA, and they can be converted to fat storage. So you don't have to memorize any of this stuff right now, but it is good to get a little bit wet, if you will, before our next lecture on metabolism. So amino acids, again, they can be used for energy, glucose, or lipids, which you can convert those to cholesterol, bile acids, and many things, lipid storage or energy. Okay, digestion and absorption. The change in a protein shape and the consequent loss of its function is brought about by heat, agitation, pH, either acid or base, will denature a protein, alcohol, heavy metals, and even other agents. Now, what I mean by denaturing is, this is the tertiary shape I was speaking about earlier. It's conformational, it twists and folds. What happens is any of these agents will start to unfold this and lead to a denatured protein, which makes it more accessible to proteases, protein digestive enzymes. So protein digestion. In protein-containing foods, the polypeptide chains are first catabolized into shorter chains into tripeptides, then to dipeptides, and lastly into amino acids. Proteins undergo some denaturing when cooked, so they start to unfold by the heat or the acid, and they start to unfold there. In the mouth, chewing moistens proteins containing foods and mixes them with saliva for swallowing. In the stomach, the G cells, this is the gastric gland. We went over this in digestion. This is the gastric gland, and this is the gastric pit. This is the lining of the inside of your stomach. This is the lining, and you have these pits that come down in here, and you have a lot of glands secreting proteases. So in the stomach, the G cells, down here, the G cells, down here in the lower part of the gastric pit, they secrete gastrin. And that is a hormone that will activate both the parietal cells and the chief cells. Now, the parietal cells of the gastric gland, they secrete hydrochloric acid, which starts to denature the protein. So in the stomach, protein denaturation starts further than cooking. And the chief cells, they secrete pepsinogen. So the G cells secrete gastrin. The gastrin stimulates both the parietal and the chief cells to release hydrochloric acid, and the chief cells will secrete pepsinogen. And the pepsinogen is converted into pepsin by the hydrochloric acid. And pepsin it breaks the polypeptide bonds of long polypeptides into shorter polypeptides and some amino acids. In the small intestine, cholecystokinin, a hormone secreted by the intestinal epithelium, stimulates the pancreas to release trypsinogen, chymotrypsinogen, and procarboxypeptidase. Now, then enteropeptidases, also secreted from the intestinal epithelium, convert trypsinogen to trypsin. And trypsin converts chymotrypsinogen to chymotrypsin and procarboxypeptidase to carboxypeptidase. Now, let me show you what I was just talking about. So you bring in a protein into your stomach, and the gastrin will stimulate the pepsinogen and hydrochloric acid to release from the chief cells and the protocells. And then the hydrochloric acid will start to denature this protein, unfolding it. And then the hydrochloric acid will also convert the pepsinogen to pepsin. And that pepsin will break these long polypeptides into shorter polypeptides. And as this protein goes down further, cholecystokinin released from the intestinal epithelium will stimulate the pancreas to release trypsinogen, chymotrypsinogen, and procarboxypeptidases. And then the enteropeptidases, they will convert trypsinogen to trypsin, and then trypsin will convert the chymotrypsinogen to chymotrypsin, and procarboxypeptidase to carboxypeptidase. Now, a little further. These proteases, the protein digesting enzymes, are specific in the bonds they break. Now, at this level, we don't have to memorize what bonds each one of these proteases break down. Basically, I want you to recognize that pepsin, trypsin, chymotrypsin, and there are several peptidases here. So trypsin, it breaks the peptide bonds of lysine and arginine. These proteases are specific in the bonds they break. So they will act on specific peptide bonds. Chymotrypsin breaks the peptide bonds of phenylalanine, tyrosine, tryptophan, methionine, asparagine, and histidine. Carboxypeptidases, they break the bonds of amino acids from the carboxyl end. And intestinal peptidases, these are the tripeptidases. These are the peptidases that are released from the intestinal epithelium as these proteins come down into the small intestine. Most specifically, the small intestine, the duodenum, releases all these receives all these pancreatic enzymes and the epithelium releases some more of these proteases, the intestinal peptidases, and then the absorption is more in the jejunum. So anyway, the intestinal peptidases are the tripeptidases. They break the tripeptides into dipeptides and amino acids. The dipeptides break dipeptides to amino acids, and aminopeptidases break oligopeptides from the amino acid end. And I have a picture, I'll show you this a little bit in another slide. Elastase are enzymes that break the bonds of elastin, which determines the composition or the properties of connective tissue. And collagenase are enzymes that break the bonds of collagen. So here in a more of a picture representation I can show you. Protein absorption in the small intestine. There are many specific protein carriers that transport amino acids. And some dipeptides and tripeptides cross the microvilli, the apical surface. Now this is the intestinal lumen of the digestive tract, and then it goes into the, it's absorbed into the body. But this is the apical membrane. Basolateral, apical and basolateral, I'm sorry. Amino acids that are not needed for energy or synthesis of other compounds by intestinal cells cross the basolateral surface. So some of these amino acids that are taken up into our intestinal cells, the amino acids may be needed for those cells. If they're not needed, then they'll cross into the basolateral side of the cell and go into our body. And the fluids that enter the capillaries and drain into the portal vein to be transported to the liver. Now here we have, like I was showing you, these are oligopeptides. These are 3 to 8 amino acid residues. They can be converted by the amino oligopeptidase, and they can be broken down to di and tripeptides, which can be broken down by aminopeptidases and broken down into amino acids, and then these protein channels will active transport, amino acid carriers, will transport these proteins into our body. Also, there are carriers that will transport the di and tripeptides into our body also. So proteins in foods. Like I said, you can find proteins in any one of the food groups: fruits, vegetables, grains, dairy, and of course proteins food group. Grains such as bread, cereals, rice, and pasta, they have around 2 to 3 grams of protein per serving. Vegetables such as yams, sweet corn, and broccoli, and there are many other vegetables, they'll contain around 2 to 3 grams of protein per serving, pretty much the same as grains. Now fruits, they have a little less, such as oranges, bananas, and peaches. They have a little bit of protein, less than a gram per serving. And dairy, such as cheese, milk, and yogurt, they have 8 to 10 grams of protein per serving. And the protein group, such as meat, fish, eggs, poultry, dry beans, nuts, they have around 7 grams per serving. Protein quality. Simply put, protein quality is dietary protein intake that promotes growth in children and provides maintenance for adults. Protein quality is basically determined by the body's amino acid requirements and the body's ability to digest, absorb, and incorporate them into the needed proteins for our bodies. The digestibility of protein depends on whether the protein is either plant, with digestibilities at 70 to 90%, or animal, with digestibilities at approximately 90 to 99%. And it depends also on the combinations of foods consumed with the proteins. So protein quality, again, dietary proteins that consist of all 9 of the essential amino acids in sufficient quantities to meet our body's requirements are considered high quality or complete proteins. And you'll find those in meat, fish, poultry, milk, eggs, and cheese. Dietary proteins that lack 1 or more of the 9 essential amino acids are considered low quality or incomplete proteins. Plant proteins lack 1 or more essential amino acid. Soy protein is the highest quality of plant protein because it contains all of the 9 essential amino acids. Now, there's an all or none principle with protein synthesis. We need all the essential amino acids to be available in our diet, or we will be able to use none of the essential amino acids, and that is considered the all or none principle. Now, it is this, but our body, if we don't have a specific amino acid, we will convert, like I was telling you, transamination, convert one amino acid onto a keto acid and get the amino acid we need. But that is, it's limited. That part of protein synthesis is limited, and we really don't want to start catabolizing our proteins. And that's why we come up with this all or none principle. Protein synthesis must have all of the 9 essential amino acids for the production of proteins. When any one of the 9 essential amino acids have been depleted in the body due to an incomplete dietary protein intake, protein synthesis will stop to the point that the body will start breaking down proteins for amino acid in its prioritization. So that leads us to a limiting amino acid. So whenever you come to an amino acid that is the least amount you have in your dietary intake or in your amino acid pool, that becomes the limiting amino acid. And that limiting amino acid will be the one that stops the protein synthesis or starts you to start breaking down other proteins in your body to meet that protein need of a specific protein that your body is trying to build. So here I have a graph for a limiting amino acid. And the limiting amino acid, is the essential amino acid that is the most deficient in the amount needed to promote protein synthesis. And that is the limiting amino acid. Here we have legumes. And this is they are deficient in methionine. That is the most deficient amino acid in legumes. And to a lesser extent, tryptophan. So methionine and tryptophan are the least amino acids, but methionine is the limiting amino acid in legumes. So generally, if all you're eating is legumes, and you have, once you hit, where does this lead to? About 25%, and I just made this up just to represent this limiting amino acid. But methionine and tryptophan are the deficient amino acids in legumes. Now, once you get to about 25% of this methionine, you have no more methionine, and that limits your protein synthesis. You can no longer continue building proteins because you have an amino acid that you're lacking. So each protein requires the sequences of amino acids, and it may require the methionine. So there's a way to complement that, and that is with complementary proteins. Low-quality protein foods, such as plant proteins, that are combined to meet the essential amino acid needs of the body. So here we have grains and legumes. Grains is represented in white, and the legumes are represented in blue again, and the legumes, again, methionine and tryptophan. But now we have grains that will complement this deficiency in methionine, so now you're getting a complete protein. Animal proteins, they are animal foods. They contain all the 9 essential amino acids. And they contain vitamin B12, iron, zinc, calcium, and fish fatty acids contain omega-3 fatty acids, fish oils. The top 5 protein foods in North America are beef, poultry, milk, white bread, and cheese. North Americans consume approximately 70% of their protein intake from animal sources, from animal foods. Worldwide, it's approximately 35% of protein comes from animal foods. Plant proteins, they are plant foods. They are low in complete proteins, but are high in vitamins, minerals, phytochemicals, and fiber. They contain low to no cholesterol and limited amount of saturated fatty acids. Okay, the health effects of proteins. Plant proteins and health. Plant proteins, they promote heart health because they have little to no cholesterol. They have no trans fats. They have little saturated fats, and they're high in monounsaturated and monounsaturated and polyunsaturated fatty acids, and fiber and phytochemicals. Plant proteins are also considered good for cancer fighting because of the antioxidants they have. And antioxidants basically are, what they do is they bind with free radicals. And a free radical is some type of atom that has a free electron, a single electron that's looking to bind with another electron. So what it does is it will attack cell membranes, DNA. It will attack cells that we don't want to attack to. It will attack atoms that we don't want to. So antioxidants will bind with that, and there are many different antioxidants. We'll go across those later in vitamins. But they act as cancer-fighting and phytochemicals. And phytochemicals, they also act as antioxidants, and some act as hormone-like compounds. And also, plant proteins, they give us better glucose control through a high fiber content. The fiber will regulate the amount of glucose that is absorbed into our gastrointestinal tract. So it regulates the process of intake of glucose. Okay, the types of diets there are. There are vegans or vegans. That means they consume no animal products. Fruitarians, it's fruits, nuts, seeds. They eat honey and some vegetable oils. They don't eat any animal products, vegetables, or grains. Now, this is not really a good diet to have. You can come into many nutrient deficiencies with a diet like this. Lacto-vegetarians, this is a vegetarian diet that includes dairy products. And lacto-ovo-vegetarians, these are vegetarian diet that includes dairy products and eggs. Lacto and ovo is eggs. And there are concerns for infants and children on a vegetarian diet because you don't get a good quality iron, vitamin B12, vitamin D, and calcium that we see in dairy products. So you have to be really good at meeting complementary proteins and meeting the right needs to meet the needs for children when you're vegetarian. Okay, the nutrient concerns for vegans are vitamin B12, again, iron and zinc. These all come from meat products. Calcium from dairy and omega-3 fatty acids from fish oils. High protein diets. Plant foods are low in complete proteins but are high in vitamin, phytochemicals, and fiber. Now, animal protein is high in saturated fat and cholesterol. Excessive intake of processed red meat is linked with colon cancer. There are numerous studies out there that connect excessive intake of red meat with colon cancer. They come up with several different reasons, but they're not exactly sure right now. And high protein diets put a burden on the kidneys from the increased urea excretion. The kidneys have to do the nitrogen balance, so it has to work harder to excrete the liver has to process the ammonia, and then the kidneys has to excrete the urea in the form of urine, so they have to work harder. And high protein diets may increase calcium loss in the urine because protein attracts water, and the excessive water loss will also probably lead to the calcium loss. So protein recommendations. The RDA for protein, it promotes an equilibrium of nitrogen equilibrium, and that is the 0.8 grams of protein per kilograms of healthy body weight. And a quick example I have here is a 170-pound man or woman, and to convert this into the metric is you divide it by 2.2 kilograms per pound, and you come up with 77.28 kilograms. And then you'll take that, the 77.28 kilograms, and multiply that by the RDA for protein, which is 0.8 grams of protein per kilogram, and then you multiply that, and you get 61 grams of protein per kilogram of healthy body weight. So a 170-pound person will require 61 grams of protein per day. And you have the AMDR, which is the acceptable macronutrient distribution range, which is 10 to 35%. So protein recommendations, they are increased by approximately 10 to 15 grams per day for pregnancy, and endurance athletes, they may need approximately 1.2 to up to as high as 1.7 grams per kilogram of healthy body weight. And we don't recommend anybody that intakes anything greater than 2 grams per kilogram of healthy body weight. The RDA is set to where you will consume about 8 to 10% of your total calories from proteins, that 0.8 grams per kilogram. You'll end up consuming about 8 to 10% of your calories from protein. And most of us eat more than the RDA for protein. Excessive protein cannot be stored as protein, excessive intake of protein. So we can't store that. Well, if we meet our needs with the glucose and the lipids, then that protein will be stored as fat, or we'll break it down, transaminate it, break it down, deaminate it, and excrete it as urine, and use the carbon skeletons to build other compounds in our body. So the Dietary Guidelines for Americans, 2010, they increase intake of fat-free or low-fat milk and milk products. Choose a variety of protein foods such as seafood, lean meats, poultry, eggs, beans, soy, and nuts. Increase amount and variety of seafood consumed. Replace protein foods high in solid fats. Okay, protein energy malnutrition, excuse me. Okay, what we have here is two different pictures of children. And each one of these children are in protein energy malnutrition. One looks completely emaciated. He looks like he's completely starving. And this child looks like he's a little better off, but actually he still has a lot of challenges himself. This bloated looking feeling is edema. And the emaciated look is called Marasmus, and the child is actually wasting away and dying. And this kwashiorkor, this is, like I said, it is a bloated look in the child. So let me go over each one of these. Kwashiorkor, that is a Ghanaian word that means the sickness the baby gets when the new baby comes. So basically, whenever a mother is breastfeeding, they have a baby that is breastfeeding, and then it'll breastfeed for a period of time, and it's probably getting some good proteins. But then she'll have another baby, and that first baby is displaced from the breast milk. Then the second baby will start receiving the breast milk, and that baby that was displaced from the breast milk may end up with kwashiorkor. And that is in populations of third world and developing countries living below poverty. It is a protein deficiency accompanied with infections. It occurs in late infancy because the child initially is receiving protein, and late infancy between 18 months to 2 years. And the first child's diet changes from the breast milk to starchy foods. The staples in Africa are sorghum, millimil, and maize. So these these starchy foods, they are like 90% starch, fibers, a lot of fibers. So you're not getting a good quality nutrient intake. The fiber is filling them up, and it has very low fat and almost no protein unless they're mixing some type of milk in with it to create the millimil. So again, the first child's diet changes from breast milk to starchy foods, and that's when he's displaced from the breast milk and they start eating these porridges of millimil or sorghum or maize. And the decreased energy consumption is accompanied with infections that compounds the need for calories and protein. Insufficient protein intake leads to loss of fluid uptake into the vascular system, causing kwashiorkor's hallmark clinical symptoms, edema. So when you're not getting, the protein intake, as I was talking about early, then the fluids will seep into the capillary beds and start causing edema. Marasmus, that is a Greek word that means wasting away. Most notable in countries under famine conditions, they're not getting any food. So it is a protein and calorie deprivation. It occurs in early infancy between 6 to 18 months. And that's when the baby is very young and they're just not getting breast milk. Their mother is probably not eating also, and she's probably not producing any breast milk or high quality breast milk. The child does not receive breast milk or stop breastfeeding early. The body deteriorates, saving the heart, lungs, and the brains for last. And the digestive enzymes are limited, and the GI tract starts to deteriorate. So you don't have the proteins to build the digestive enzymes, and you don't have the proteins to maintain the digestive tract. So when the digestive tract starts to deteriorate, you also have a decreased absorption. Progressive deterioration of subcutaneous fat and muscle. The child is emaciated, skin and bones, it looks skin and bones, and the child is dying. So here is a comparison of the characteristics of kwashiorkor and marasmus. Here in kwashiorkor, it's late infancy to early childhood, and marasmus, it's in infancy, they are starving, and the young babies, they are not getting the protein or the calories. In kwashiorkor, it's a protein deficiency accompanied with infections, and in marasmus, it's protein and calorie deprivation. They also have many infections. And in kwashiorkor, it's acute protein energy malnutrition. Acute because the baby was displaced from the breast milk. And in marasmus, it's chronic protein energy malnutrition because it hasn't been getting the protein or the calories. So in kwashiorkor, it's the 60th to 80th percentile weight for age. And in marasmus, it's less than 60 of the percentile weight for age. In kwashiorkor, there is edema. In marasmus, there is no edema. In kwashiorkor, it is the hallmark, the swollen abdomen from ascites, the fluid in the abdomen, and also you will lead to a fatty liver disease. And in marasmus, the child is emaciated. So this one looks a little plump, and this one looks like it's definitely starving. So in kwashiorkor, there's decreased muscle mass. In marasmus, there's decreased muscle, fat, and tissue that's wasting, like that's saying in the gastrointestinal tract. And in kwashiorkor, there is decreased appetite, whereas in marasmus, the child has an appetite, he's very hungry, a voracious appetite, but he's not getting any food. In kwashiorkor, it's lethargic, irritable, and there's fatigue, whereas in Marasmus, he's mentally alert and has anxiety and also irritable. Kwashiorkor, there is diarrhea. And in Marasmus, you have chronic diarrhea when you're starving so much and you start to get some kind of fluids in, but your gastrointestinal tract is deteriorating, the fluids come out. So there's chronic diarrhea and vomiting. In Kwashiorkor, there's dermatitis, lesions, and pigmentation changes. And similar in Marasmus, there is this dry skin, loose. The difference here is it's loose. It hangs in folds on the buttocks and underneath the arms. The skin is actually loose. So, and in Kwashiorkor, the hair is dry, thin, and it is sparsely populated. Whereas in Marasmus, the hair is dry, it loses color, and it curls. And here is a link if you want to get more information on Kwashiorkor or Marasmus to the National Institute of Health.